Hello everyone, let's do this paper IGCSE paper 6, that is alternative to the practicals, and this is October November 2022. So the question number 1 is about some students investigated the effect of hydrogen peroxide concentration on the activity of the catalase. And catalase is the enzyme that is found in the yeast cell, and it is used to break down of the hydrogen peroxide to form water and oxygen. So actually the reaction is hydrogen peroxide, and we have the catalase enzyme. It can break down into water, break down the hydrogen peroxide into water and oxygen. So this is the reaction, and the student compare the activity of catalase in two different concentration of hydrogen peroxide solution, and during that they also change the temperature during this reaction. They also use a yeast solution as a source of the catalase. They also use in the step 1 they use aluminum foil and wrapped around the outside of the test tube. So aluminum foil is used around the test tube to prevent the heat loss, and this is the test tube A, and there is another test tube B. Aluminum foil was wrapped around the outside of the second test tube and it is called B. They also use this syringe to measure the volume of the gas, that is oxygen, and the temperature of hydrogen peroxide solution in A was measured was 20 degree at 0 minute. Step 5, the yeast suspension was stirred, and 1 centimeter cube of yeast suspension was then added to the hydrogen peroxide solution in the test tube, that is called A, and use a stop clock to measure the time. And step 6, the temperature of the hydrogen peroxide solution in test tube A was measured for every minute for the 5 seconds. And step 7, the syringe was used to put 5 centimeter cube of hydrogen peroxide solution and 5 centimeter cube of distilled water into the test tube B. So the step 8 says that 4, 5 and 6 were repeated using the test tube B, and here we can see the measurement that record in the first 5 minutes, and the second one they just show us the test tube B and the measurements. The question is prepare a table and record the results that show in step 4, the figure 1.1 and 1.2. So if you see here the first figure, they show us the time that start from 0, 1, 2, 3, 4, 5 minutes, and on this side they give us the temperature. Same thing they show us in the figure A, the test tube A, but that start from 1 minute, 2 minutes, 3, 4 and 5, and on this side we have the temperature. When we are going to make a table, we'll use the time, and don't remember to the units, that is the second, and for temperature the unit is degree centigrade, and because we have two test tubes A and B, and the time started 0. The same temperature is for A, but they didn't mention in here, but as it started the temperature is so here is 0 minutes, and the same for the B20, and we can write the other measurements. The minute 1 is 24 degrees, same for the B, 24, 28, 26, 31, 28, and at 4 minute the test tube A, the temperature is 33, B is 39, and 5 minute the temperature in test tube A is 34, and in test tube B the temperature is 29. So that is our table. Very simple. Question 2. Using the result, identify the highest temperature and lowest temperature in each test tube. So use these values to calculate the maximum temperature change. So we measure the maximum temperature change in each test tube. So if you see in the maximum temperature in test tube A, I calculate is 14, because 34 is the maximum and minimum is 20, that is 14. If you do this same for the test tube B, the maximum is 29 and the minimum is 20, that is 9 degree. The next question was state a conclusion for the result. So the conclusion, if you see here, if the concentration of the hydrogen peroxide increase, the activity of catalase enzyme also increase. Next question, state the variable that was measured in this investigation. Dependent variable. What we measure in this investigation is the effect of temperature. That is what we measure. So as the temperature increase, the activity increase, and after optimum temperature, the activity will be decreased, because the high temperature can denature the active site of the enzyme and there will be no increase. The part B, the test tube were covered in foil in step 1 and step 2. So you predict the effect on and explain the effect of the result if it is not covered with the test tube with foil. So the foil was used to around the test tube. This is a test tube and there is foil that we cover this test tube. So if it is not used, so it can lose the temperature. The effect on the result, if room temperature is lower, as without foil there will be heat loss. So foil is used to protect from heat loss. The next question, 2 variables that other than covering both test tube with foil that were kept constant. So there are a lot of other variables, the volume of the solution or the concentration, type of yeast, volume, anything you can write. The next question say, in a different experiment, the student investigated the effect of pH on the breakdown of hydrogen peroxide and by calculating the rate of oxygen production. State 2 measurement that the student would need to take to calculate the rate of oxygen production in centimeter cube per minute. So because hydrogen peroxide break down into water and oxygen, so to measure the volume of the gas, the 2 measurements the student must take is the volume of the oxygen gas and the time, how much volume is produced and how much time in how much gas is produced. These are the 2 measurements that student must need to take to calculate the rate. If the volume is high, the rate is faster, and if the time is less, in the less time, if the more gas is produced, it means the rate is high. The result of investigation they show us in the next table, and the first trial, the second trial, and the third trial, and there are different pH on one side, and here they show us the average rate of oxygen production. You can see in this table that as the pH increase, the rate of production also increase, and if the pH increase too much, the rate decrease. And what is the question? What is mean by anomalous result? Anomalous is mean different result that do not fit with the pattern. So if you see at pH 8, so trial 1 is the rate of oxygen production is 27, but trial 2 we got 16. The 16 here is the anomalous result, because 28, 27 is not a very big difference. So this 16 is the anomalous result. A result that do not fit with the trend is called anomalous result. The next question, describe how the student calculated the average for pH 8. So by calculating pH 8, you can just simple add the trial 1 and trial 3, that is 28, and take the mean value. So they did not use the anomalous result in the calculation, or they just add the other two trials and divide by 2 to get the average value. The next question is, using the data in table 1.1, plot a line graph in the grid to show the effect of pH. Effect of pH here is important, and the average rate of oxygen production. So on one side you can write the average rate of oxygen production, and the unit is very important, centimeter cube per minute. And on the x-axis you can write the pH, and you can see here that by these two are important. pH on the x-axis I write, and the average rate of production right on the y-axis, and average rate of oxygen produced. So when the pH is 5, the average rate of production is 7.7, and the pH 6 is 16, 15.3, 15.3, and the next one at 7, it is 27.3, and at pH 8 is 27.5, and the pH 9 at it become 5. So you can simply just add this and make a line and connect the lines. The next question is, describe the effect of pH on the rate of production. So I already explained this, that the rate of oxygen production increase and then decrease, and the optimum pH is 8. After the pH 8, the rate of production of oxygen decrease, because the high pH can denature the enzyme and it will no longer bond with the substrate, and there will be no more reaction. The next question is, the student wanted to obtain a more accurate value for the pH, at which average rate of production was highest. Suggest what further investigation work the student should do. If he want to obtain more accurate value of the pH, so he should do this investigation again and do this with more smaller intervals of pH, between pH 7 to 9. Then he can obtain more accurate values of pH. If the distance of pH is not very big, as in here, if you can see this 5, 6, 7, so further he can increase the pH a little bit, 5, 5.5, or 6, 6.5, like this. The next question is, they show a photo micrograph cross section of the stalk of celery, and the question is show us to draw the diagram of the celery stalk. Remember, when you are going to draw this diagram, just remember just few things, that you should not shade anything. Your lines should be clear and more continuous. Okay, so it should be, lines should be more clear, plus they should be more continuous. And the next thing that is more important is the image should be larger image. The space they provide, you must use that, and you should not draw a small image, and do not draw any shade anything. And the features that are prominent, for example, here you can see these dark circles, so these are important. So we have 1 2 3 4 5 6 7 8 9 10 11 12 and a little bit 13, and the margins are very important. So we will draw like this margin, this edge is important, and this one, the next here, this, and same like here. That is like I draw like this. And the next one, they say label one vesicle bundle on your drawing. So this one you can just choose any one, that is vesicle bundle. The next question, the actual width of the cross section is 27. So measure the length of P to Q. So I have measured this distance. You can use your ruler, and you try remember the unit, that is millimeter. So the length of P to Q is 78. And the question next they ask you is to calculate the magnification. So the magnification M, so M formula is length of the image, our actual image. So there is 78 over 27, so that is 2.9. And we because give your answer as a whole number, so the whole number we must have to write 3 is our whole number. The next question is movement of water uptake by the silvery stalk can be investigated by placing a silvery stalk in a solution of dye like this. This is the dye, and we have put silvery stalk in there, and we will measure how much dye that will travel along the silvery upward, and then we measure that. And question it says plan an investigation to determine the effect of air temperature on the movement of dye up a silvery stalk. So you know that the temperature will affect the movement of the dye up, because when the temperature is high, there will be more water loss, more transpiration. The leaves of the stomata, the leaves, and they have stomata, will be more open, more water loss, and the xylem, in the xylem, the water will move upward. So in different temperature, if the temperature is low, the transpiration rate will be low, so the distance will be less. So the first one is I have use 5 silvery stalks different, and we will put each in different air temperature. So you can add more different temperature. First is you can add like 30 degree, 40 degree, 50 degree, 60 degree, and that's our independent variable. Use same type of plants, same humidity, that is control variable, same intensity of light, and same wind speed. And then we measure the distance that is moved by the dye up in the stem, because in different temperature the distance traveled by in the silvery stalk will be different. The high temperature, there will be more distance covered, and then we'll cut the pieces of silvery, and the distance you can choose any, like I write here 0.5 centimeter cube, or maybe 1 centimeter cube or 2. And then and the safety, you must have to write a safety measurement that we must uh, take during our investigation, because we are cutting, so we must cut on the cutting board and must cut away from the body. So do not cut yourself, and use the goggles and gloves, and repeat the investigation. So all these steps are very easy, you don't need to remember, just go step by step, and complete and very easy. There is another method they ask you to describe that can be used to find out the samples. If the sample of plant tissue contain reducing sugar, if you have any samples or the tissues of the plant, and if you want to do experiment to check if they have any reducing sugar, first one is we have to crush in more and small pieces and grind them there, so the surface area can increase, and then we mix with the water to make a solution. So in that solution we will add more Benedict solution and heat it. So if there is any reducing agent present, the color will change from orange. The color will change to orange and red, because the question have 3 marks. So you must have to write at least 3 steps: grinding, crushing, and make a solution, add the Benedict solution, heat it, and the color change. So that is our paper. If you guys have any question, you can comment and I will love to answer. Thank you. Goodbye.